Thank you so much for joining back. In this lesson, I'll be providing an introduction to nucleic acids. Nucleic acids are a biomolecule we'll be talking about a lot when I go over gene expression, cell division, and protein synthesis. This is because nucleic acids are used by all life to store their genetic code or genetic makeup. But before I go into the purpose of nucleic acids, let me first introduce their structure. Nucleic acids are made up of the following elements. You have carbon, oxygen, hydrogen, nitrogen, and phosphorus. And phosphorus is one of the elements that we haven't seen before in carbohydrates and proteins. So I've drawn out a nucleotide, which is the monomer of nucleic acids. So let's label this nucleotide. And when you look at the structure, I'd like you to take a minute and think about the side groups that can be found in this. So now that you're back, I'm going to be going over the structure and the side groups that are found in nucleic acids and specifically in their monomer nucleotides. First, let's start with their phosphate group. You may notice that the phosphate group is charged and it's negatively charged, which will be important later on. The next part, and this is the nitrogenous base. And the way you can remember that it's a nitrogenous base is the high amount of nitrogen that is found in this structure. Again, I'm not drawing the carbon atoms in this structure, and instead those are the corners that are not labeled. The final part, and this should be familiar, is actually a sugar. So this is a sugar. Specifically, this would be ribose, and I've gone over this in the past, which can be found in ribonucleic acid. The section made up of the nitrogenous base and sugar is called a nucleoside. So this structure is the nucleoside, and the nucleoside, in addition with the phosphate group, makes up the nucleotide. Again, going back to this, an important thing to note about the nucleotide is that it's negatively charged, which is derived from the charged phosphate group. This is important as the charged nature of the monomer allows DNA to be tightly packed on positively charged histone molecules in the nucleus and to be isolated when we extract DNA out of the cell. Going further, nucleotides have 5 common nitrogenous bases that I've drawn out for you below. You have adenosine, cytosine, guanine, thymine, and uracil. And I'm going to star uracil. And the reason I'm doing this is because there's actually 4 nitrogenous bases found in DNA. And those are the 4 to the left: adenosine, cytosine, guanine, and thymine. In RNA, uracil actually replaces thymine, and so it's not actually found in DNA. The structure, as you can see, also differs. So the nitrogenous bases are not all uniform. There's 2 classes. You have pyrimidines. You have pyrimidines. And you have purines. So again, I'd like you to take a second to try and see the difference between these 2 subclasses, and when you come back, I'll be giving you the answer to this question. So now that you're back, the purine, if you notice, is the larger nitrogenous base, and it's made up of 2 rings, while the pyrimidine is made up of single rings. And so cytosine, thymine, and uracil are actually smaller. This actually factors into when we talk about DNA, which is a double-stranded polymer. And this is because adenosine and thymine are hydrogen bonded to each other, and guanine and cytosine are hydrogen bonded to each other in the DNA structure, which allows us to create the double helix I'll be talking about later. So you would have a purine go with a pyrimidine in these structures. And so again, the difference between these two is that this has 2 rings and that this has 1 ring. So now that we've covered these monomers, I'm going to be talking a bit about DNA and RNA. So let's choose different colors. You have DNA and RNA. Cellular RNA ranges in length from less than 100 to many thousands of nucleotides. So you have 100 to 1,000 or greater nucleotides, while DNA can be several 100 million, so 100 million nucleotides long. And so this factors into what I was talking about before with histone molecules, where the DNA is extremely long. And so to pack it within the tiny cell, it's necessary to bind it to the histone molecule. And so it helps that the DNA, which is negatively charged, is attracted to the positively charged histone. DNA is found in a helical double helix shape. So you have this double helix and it winds. So you have this structure that you've commonly seen. And the way that it goes is, and I'll draw this out as just not in the double helix, but just straight strands. It goes from a 5' end to the 3' end, and the 3' end to the 5' end. The 5' to the 3' is actually the parts of the sugar. And so when you look at a sugar, and I'll be drawing this underneath the RNA, because RNA will have a bit more space. The sugar has its 5' here, and that would be the 5th carbon. You have 4' here, 3', 2', and 1'. And this kind of hints to where these nucleotides are linked. So each nucleotide links from its 3' to the 5', and it goes downwards. Cool. So RNA is a bit more simple, and it only has 1 helical strand. So you only have 1 helical strand. Again, we'll be diving deeper into the functions of RNA and DNA, but as a summary, these nucleic acids store the information about the makeup of an organism, down to the smallest protein and the mechanisms for regulation for the entire body. So in summary, nucleic acids are made up of carbon, oxygen, hydrogen, nitrogen, and phosphate, or phosphorus. Nucleotides have a phosphate side group, it has a sugar, and it has a nitrogenous base. These nitrogenous bases are adenosine, guanine, cytosine, thymine, and uracil, with the first 4 found in DNA and the first 4 excluding thymine and instead having uracil found in RNA. And they're split up into purines and pyrimidines based on the rings or the amount of rings that they have. In DNA, adenosine and thymine are bonded together, hydrogen bonded together, and cytosine and guanine are hydrogen bonded together. And DNA can have 100 million nucleotides, while RNA ranges from 100 to 100,000 or more nucleotides. Again, just as a summary of the purpose of nucleic acids, they store the genetic code or genetic makeup of an organism, and we'll be talking about this a lot more extensively when we talk about the gene expression and genetics and protein expression.